Hello class. As we learned in lecture, Edward Jenner made a vaccine against smallpox. He started by injecting the young boy with material from the cowpox pustules. The boy got sick, but he got better. This led to the conventional vaccines that we still use today. They work, but this technique has its problems. The technique most popularly used for the past decades is, in a nutshell, isolating, inactivation and then injecting weakened or dead microbes into people. However, this technique cannot be used with pathogens that can't be cultivated in vitro, which means in the lab. This process is also very lengthy. It can sometimes take up from 6 months to 5 years, depending on the pathogen. But now, a newer and a better approach has been given rise. Reverse vaccinology, the genomic approach. This picture shows that the traditional vaccinology technique can sometimes take more than 3 years, while reverse vaccinology can take 1 to 2 years. Reverse vaccinology is the use of a microbe's genome, or part of its genome, to create a vaccine. Certain surface proteins trigger immune responses, and if the right immune response is created, that protein can then be used for a vaccine. Scientists have moved past just mapping out bacterial genomes, and have started using these techniques on viruses as well. Supercomputers are used to identify parts of the pathogen's genome that will encode for surface proteins. It is thought that surface proteins contains the antigens because it is accessible to the body. The gene with antigen properties is isolated from the rest of its genome and spliced into a plasmid. Cloning of the gene coding for the antigen in large quantities is needed to characterize and produce the identified antigen. The plasmid is then injected so that host cells can transcribe and express the gene. The host will now produce the antigen for that particular antigen. Because computers are used to detect these proteins, this process is a lot faster than the conventional vaccine approach. This picture is a visual of the supercomputer selecting the surface proteins to see which ones would be expressed. The proteins are then placed in E. coli, and the expressed proteins are inoculated into mice. The second picture shows the removal of a pathogen's genome and splicing it into a plasmid. This altered plasmid can then be used for one to start generating antibodies against the targeted pathogen. This approach was first used on a group B meningococcus and was one of the first successful reverse vaccinology application. Fragments of the DNA were screened by computer analysis. It was predicted that 600 genes would code for surface-exposed proteins. These genes were then cloned and expressed in E. coli and were replicated. The successfully expressed genes were purified and injected into mice to immunize them, and it was found that 25 surface-exposed proteins induced antibodies. This is the base of the meningococcus B vaccine. Another approach of reverse vaccinology just uses plasmids. After isolation of the protein with antigen properties, the polymerase chain reaction is used for replication. A human polymerase DNA fragment is inserted into a pathogen's plasmid and would then be cloned. Segments of the DNA would be removed so that it can be used in humans. After further synthesis of the DNA, incubation and being placed in a centrifuge, a vaccine is now ready. This picture shows the polymerase chain reaction and the centrifuge used to make vaccines. Benefits of reverse vaccinology. The conventional methods are more time-consuming than making a reverse vaccine. The reverse vaccines, especially the ones done with plasmids, have no contact with any pathogens, only the genome. This leads to fewer side effects like sickness. These techniques have been successfully used with hepatitis C, Group B streptococcus, and Group A meningococcus.